In this topic, we're going to have a look at movement in and out of cells, focusing on diffusion. So by the end of this topic, you should be able to answer the questions: what is diffusion? What are the factors that affect the rate of diffusion? And what is facilitated diffusion? Now, as you remember, all cells are surrounded by a partially permeable membrane that controls what substances can enter and exit a cell. So a cell needs to be able to import the substances it needs to survive and export waste materials and other substances that are needed outside the cell. So there are several methods by which substances such as molecules and ions can cross the cell membrane. These include diffusion, osmosis, active transport, and bulk transport. Today we're just going to look at diffusion. So what is diffusion? We say that diffusion is passive transport. It's the net movement of particles down a concentration gradient from a region of high concentration to a region of low concentration, either in a liquid or gas. So here you can see the pink molecules are moving from the left, where they're in high concentration, to the right, where they're in low concentration. So molecules will passively diffuse from an area of their high concentration to an area of low concentration until an equilibrium is achieved. Now, since all movement requires energy, it might be confusing to describe diffusion as passive. What I mean by passive is that the energy comes from the natural inbuilt motion, which is the kinetic energy of the particles, rather than from an outside source. So what are the factors that affect the rate of diffusion? You've got the steepness of the concentration gradient, surface area to volume ratio, temperature, and the nature of the molecules or ions. I want you to copy these 4 points down and then make notes as we go along. Okay, looking at the steepness of the concentration gradient. If you were to draw a concentration gradient, on the left you'd have a high concentration, and on the right you'd have a low concentration. So here you can see the orange particles are in high concentration on the left, low concentration on the right. So which way will diffusion occur? It's going to occur from left to right. So the greater the concentration difference, the faster the diffusion. So we want to maintain a steep concentration gradient. How do we do this? Well, we constantly add particles to the left and remove particles from the right. So maintaining the steep concentration gradient ensures that the rate of diffusion will be high. So where do we see diffusion occurring? Well, if you think of gaseous exchange between the capillaries and the alveoli, we need to maintain a steep concentration gradient there. How do we do that? Well, if you think of carbon dioxide, which way does carbon dioxide diffuse? It moves or diffuses from the blood in the capillaries into the alveoli. And then what's going to happen to it? It's going to be breathed out. So the concentration of carbon dioxide in the blood is high because the carbon dioxide is coming from respiring cells. It diffuses from an area of high concentration in the blood to an area of low concentration in the alveoli. How is the concentration maintained at a low concentration in the alveoli? This is because we're continuously breathing out carbon dioxide. So this maintains a steep concentration gradient. Looking at surface area to volume ratio, you should remember this from IGCSE. An increase in the surface area to volume ratio increases the rate of diffusion. So things like villi and microvilli increase the surface area to volume ratio. And the shape of an organism can also affect this ratio. Temperature. What do you think temperature will do to the rate of diffusion? So an increase in temperature will increase the rate of diffusion. So diffusion is passive and depends upon the kinetic energy of molecules. So here you can see how the molecules move in the cold compared to if you apply heat. So you're giving them kinetic energy, more kinetic energy, should I say. Oops, and there's one last one to move. Okay, then the last one is the nature of the molecules or the ions. So lipid solid molecules and very small molecules such as oxygen and carbon dioxide can diffuse directly through the lipid bilayer. What about polar substances? How do they move through the bilayer? Can they move directly through the lipid bilayer? No. They need special proteins to help them move, and these special proteins have got a hydrophilic channel. So what happens is water molecules or large hydrophilic molecules will diffuse through these protein molecules, and we call this facilitated diffusion. So facilitated means to help. So what is facilitated diffusion? Facilitated diffusion is a passive process by which molecules move down their concentration gradient through protein channels. So it's basically diffusion with protein channels. Okay, so the proteins provide hydrophilic areas that allow the molecules' ions to pass through the membrane. And then there's this word ionophore. So proteins that transport ions, hence ion, through the membrane are known as ionophores. So let's have a look at the different points of facilitated diffusion. So this requires channel proteins or carrier proteins, and the channels may be temporary, and we say that the ions or polar substances move by passive diffusion, since no energy is used. Substances can only move down their concentration gradient, and diffusion stops when an equilibrium is reached. The rate of facilitated diffusion can reach a maximum because it's limited by the number of protein channels. What do I mean by this? Well, if you've got 4 protein channels in the membrane compared to another membrane that's got 10 protein channels, which one will have a faster rate of diffusion? The one with 10 protein channels. So here you can see the yellow particles are moving through the protein channel by facilitated diffusion. Now there are many different types of proteins. The two we're going to look at are channel proteins and carrier proteins. So what's the difference between the two? Well, channel proteins form hydrophilic channels across the membrane. So they allow charged substances and water-soluble molecules such as glucose and amino acids to pass through them. And they're going to move from an area of high concentration to an area of low concentration. If you look here, you've got glucose, amino acids, water, salts, and ammonia moving through these protein channels, and we say that these channels are selective. So they only open in the presence of a particular molecule. So when the molecule is not present, the channel remains closed. So we say that the channel is gated. Now something interesting about these channels, if you put the protein in the membrane, what will the outside of the protein be, hydrophilic or hydrophobic? The part that's in contact with the phospholipid bilayer, it's going to be hydrophobic, and the inside is going to be hydrophilic to allow these special substances or hydrophilic substances to pass through. Okay, looking at carrier proteins. Now carrier proteins have got a binding site for specific solutes, and they constantly flip between two states. So the site is alternately open to opposite sides of the membrane. So substances will bind on one side where it's at a higher concentration, and then they'll be released on the other side where the substance is at a lower concentration. And this process only uses the kinetic energy of the molecules themselves. So can you think of some examples of facilitated diffusion? Well, the movement of ATP in and out of mitochondria, the movement of glucose, and water reabsorption in kidneys. Something that ties in quite nicely with facilitated diffusion is cystic fibrosis. Now, in your lungs, in the cells surrounding your lungs, you've got these proteins that transport chloride ions. And if this protein's not working properly, so there's a defect in the protein in the cells lining the lungs, the chloride ions cannot move out of the cells. So what happens is you get thick mucus accumulating within the lungs, and because this thick mucus is nutrient rich, bacteria is able to grow in there. Okay, in summary, what have we talked about? Well, what is diffusion? Can you remember what diffusion is? It's the net movement of particles from an area of high concentration to an area of low concentration down their concentration gradient. What are the factors that affect the rate of diffusion? There's steepness of concentration gradient, surface area to volume ratio, temperature, the nature of the molecules or ions, and then what is facilitated diffusion? Well, this involves carrier proteins or channel proteins to transport substances down their concentration gradient through the membrane. And that concludes our lesson, the end.